Josefina, please.
Thank you. Thank you very much, Gantram, and thank you for inviting me to talk a little bit of our work, which is not as usual in these kind of conferences. So I will go directly to the talk. So we have a little bit of history in genomics surveillance. We provide training for Latin America and the Caribbean for the past, I would say, 10 years through Connecting Science, through PulseNet Latin America and the Caribbean, through the Latin American Center of Study, the University of Buenos Aires. So pretty much we train more than 1,000 people around the region in the past 10 years. We started our sequencing lab in 2016, and that's to give you a little bit of context. And Liz Malbrand is the national reference lab for the Ministry of Health. So we're seated under public health. So every research we do has to be public health related. So we have been using genomics in specific projects to provide case studies to demonstrate the value of genomics in surveillance. This means that before the pandemic, and I will go after 2020 later on, all our funding has come from research. And I have to say it's quite difficult to get funded in Latin America. Philanthropic founders are not very keen to fund our regions because we are not as high, we're not as low income countries in that region. But all our funding has come from there, not from the government, which we are seated. We have been doing outbreak investigations of foodborne pathogens, sexual transmitted disease, tracking clones. We have a very nice project on tuberculosis with EBI and Samin Iqbal group, where we prove very well our case. And actually now we are doing regular genomic surveillance based on this data that we presented to the ministry. But also have been providing guidance or circulating clones of vaccine-preventable disease, and also inform on the introduction of new pandemic lineages. I'm not talking about COVID, I'm talking about cholera in this case. But in 2020, our history changed completely, like all of us. So it was the first time, just to illustrate, it's the first time our president visited our institute, even it's over 100 years and have been driven public health in Argentina. We, for the first time, have generated a genomic surveillance guideline. And this is quite important because up to then, none of our guidelines include the genomic word on it, so non-genomic surveillance was done. And why is this so important? Because these guidelines obligate the government to start funding genomic surveillance in Argentina. In this case was for COVID, which was like the obvious thing and the pandemic response that everyone was expecting, but also generate a new model for data sharing. We have to start doing the data sharing of genomic variables in our system, which is quite simple. So we only have to put something that reflects our results in a yes or no on a category basis. In this case was BOC, so BOIs. For some BOCs, we go up to lineage, but that's it. But we also generate regular reports from the Ministry of Health with genomics and metadata analysis. I will go a little bit later on our integrated system of health information, which is quite impressive in Argentina. It is linked with our ID card, as has all our health records, including our recent vaccination records. All of this was uploaded to public repositories, and we use data standard generated by FAGE, the Public Health for Genomics Epidemiology. So that's secure funding for our institute for the first time for real sequencing. So we generate a sequencing facility. So under Liz Malbrand, we have seated 14 institutes. Our is the number 14 and was created in 2021. And up to today is the biggest sequencing facility in the country, including public sector in Argentina. We have the possibility to scale up our sequencing, but also to generate a new IT infrastructure, also in the institute, and to have a robotic platform to automatize all these projects. So we went not only for pathogen genomic surveillance, but also including some human genomics as well, which I'm not going to get deep in this presentation, but if anyone has questions, I'm happy to answer later. But we have some challenges faced during this. Even we scale up very well our facility, we still have some logistic issues for sample transportation. So just imagine Argentina is a very big country in the south of the continent. So getting the samples from the provinces to the sequence facility takes a lot of time. Also, having problems in the RNA integrity. So we have some problems in getting whole genome sequence of COVID. We have also delay between the sample collection and the data release in the public repository. This was very clear in the Omicron WHO reports, where Argentina still appears in white, where we should appear in blue by the kind of sequencing we are doing. And that has to do because it takes us more than 30 days before the data is generated, before the sample is taken, until we could upload it to the public repositories. And also we have a problem of sample ID traceability. We are a federal country, so we have 24 different provinces in Argentina, each one of them with a Ministry of Health with their own decisions. And we were very openly saying, okay, but we uploaded our data, we are doing all our data in our public repositories, but it was impossible for the hospitals taking the samples to trace that data, because when they came to the National Reference Lab, they put their own ID, and that's how it was uploaded to the public repository. So we have a lot of pushback in the country because they couldn't do their own analysis, for example, in the provinces. So it's that when we secure a second amount of funding to generate the Federal Genomic Network. And the aim was there to generate sequencing capacity across the 24 jurisdictions in Argentina, using all the technologies available. And I'm talking in the early 2021, which was quite difficult to import Illumina, MI6, or getting nanopore mini ions, which we couldn't get any mini ion in Argentina for public health before this. There has been a lot of negotiation with the companies to get this in place and as soon as possible. And now we have 24 jurisdictions having the ability of sequence, all of them with nanopore technology, and 12 of them also with Illumina, so having both platforms in place. We also get, for the first time, standardized lab protocols for generating sequencing, but also for analyzing that data and upload it to the system I mentioned before. So next to your ID record, you could also get your VOC with the same protocol, which wasn't something that has happened before, because even we have a research project going on that we were part of it, the PAIS project, none of them uses standardized protocols. None for the lab part, neither for the data generation. And we try to change a little bit the concept that we have of laboratory networks, and I go a little bit deeply later. But the idea is to generate a concept of community of practice. So we are a surveillance network, but we are working directly with the provinces to generate not only the data, but also generate the capacities. So some provinces and some nodes are, for example, leading one of our some of our projects, of our new projects. It's not centralized, that leadership. And we also have that all the federal provinces, no matter if the data generates locally or by some reason generate centrally, they should analyze the data and upload it directly themselves to the national system. Also, we only have a unique ID samples to trace this data, which was a completely game changer for all of us, and have a standardized report model. And this seems something very simple, but it's not. When the epidemiologists get the reports back, they usually have different models, the trees were everywhere, data was explored in different ways, they could not understand what we are talking about, so they only read the last part of the report with the conclusion and that's it. So we have a training with the epidemiologists explaining how the report is going to be read, so they can really make some decisions over that as well. And of course, when we have this resource available, we want to go further than COVID. This is not the only reason why this genomic network was generated. And we want to develop a pathogen genomic strategy for pandemic and epidemic response that has a federal character. We wanted to address the landscape of human, animal, and environmental health. That's what I mean, that we also include human genetic projects. Considering the province capacity, the large-scale sequencing center that we generated, and all the established laboratory surveillance. And this was one of the problems. Under Malbrand, we have more than 30 laboratory networks already existed for surveillance. So we had to think, how are we going to work with them as a new network that generates different technologies? So we have meetings with all of them, or most of them, and generate some prioritize. So of course, we start working with the virus of high prevalence in Argentina, influenza, respiratory, syncytial respiratory virus, dengue and chikungunya, which are arboviruses of high prevalence in our country. And For the bacterial part, which is where I sit from, I came from bacterial genomics, which was so much complicated to deliver. We start working with 2 pathogens that are quite easy to understand. They are foodborne related, and we have a network that has more than 20 years sitting down, that it's PulseNet Latin America and the Caribbean. So we already have data sets to work with. And we start with Salmonella and E. coli. The first thing was to define and validate standardized protocols, which it was quite easy for bacterial pathogens, not so too much with the nanopore technology, but when we developed those protocols, we are already distributing to the provinces, but also to generate the new influenza and RSV protocols for Illumina and nanopore that were not that validated at the National Reference Lab. We have to define an analysis protocol for the provinces, and this has to be based on data variables, so genomic data variables. It's not that the provinces are going to analyze all the data, but the data that directly goes to the Ministry of Health in the health system. Also define number of sequences per provinces, per pathogens, according to their capacity. But not only that, according to what we want as a surveillance system. So no matter if they can generate the sequences locally, if they cannot for any reason, they have to send it centralized to get that data in place as soon as possible. We define this report with the EPI team, so not only the genomic variables, but also the standardized, more detailed reports that the provinces can also done to analyze their data locally and also to generate the national report. And then we have to define a little bit the roles in the provinces. So you have to think in a country where bioinformaticians are, and I think globally, are not available that much. So the first pushback that we have from the provinces is, should we have bioinformatician in each provinces? With the first answer, and the only one is no, because we cannot have that situation in Argentina. So what we train are genomic bioinformatic analysts that only have to go through the standardized protocol to generate these variables for the Ministry of Health. Also, we define the role of genomics epidemiology that interpret and generate the reports that are seated in the national reference labs, but also in some of the provinces lab that have some bioinformatic support behind them. And the real bioinformaticians are supported at the National Reference Lab to develop all the algorithms behind in our server. A little bit of the architecture of this new model is that any hospital in the country can generate the sample ID, that not only the nodes of the federal network, because these samples are also go for diagnosis. With these unique ID samples, this can be sequenced at the provinces lab or can be sequenced at the National Reference Lab, depending of the capacity at any time. But always the province lab must done the analysis, the basic analysis to report to the national system. All the analysis and all the databases is stored at Malvern Institute. So it's stored at the National Reference Lab. The provinces report to the system back and produce a local report if they are able. And the National Reference Lab get access to all the provinces. So each province can only get us access to their own data, but the National Reference Lab get access to all and generate the national report. And all of that is released to the public repositories with the unique ID. So each province can also locate the data. For example, not only the node of the sequencing, but for example, the hospital that generate the sample at the first time. If they want to do some further analysis, they're able to link it with all the metadata because they have access to the system for each sample that it generated locally. So the data flow goes that with any laboratory generates the case, they also upload the metadata that is linked to our health records. So if we have previous infections, that also is in the system. Then the sample ID is generated. The node generates a report that's called a surveillance event, and that's allow them to sequence. That's up to keep track a little bit of all the reagents and how many sequences is done. So even if the sequence fail, we still know that the case was generated. Then the genomic data generated at the nodes or centralized, and it's uploaded to the National Genomic Database that it's in Malvern Institute. And then the nodes analyze their variables and report to the system, and they can generate a further report. The same for the National Reference Lab. That goes again to the system. All that data is uploaded to public repositories by the provinces itself. That's the idea. We're still struggling a little bit to that, but if they're not uploaded by the provinces, we will do it for them. And then now we will see a little bit why. We have a QA program, a quality assurance program that has a little bit of PulseNet flavor because that's how we usually do our QA for genomics. So we certified each node per pathogen. So all of them are certified for COVID-19, some of them for influenza, some for salmonella, some for all of them, depending on their capacity. We have 3 minutes, so we have an annual proficiency test, and the data set of this is prepared with the National Reference Lab, and it's only for genomic data. So what I meant on helping the provinces is helping the provinces to set up their data systems because they have their laboratory management system as well, and they have to link it with this ID, unique ID system, which is quite simple for all of us, but for the provinces are not. We use the data structure contextual for COVID-19, but we're still delivering, and how are we going to use this data specification for other pathogens as well? And we also help them in translated information because the data is in Spanish to make sure they can upload it quickly to the public repositories. What about sustainability and next steps? So our IT infrastructure, we have a massive investment, still struggling. So we're thinking about cloud-based infrastructure. This is very difficult for governmental data, but we're still exploring that possibility. We still trying to help very well in relationship with our research community, in this case, the Genomic PAIS project, to make sure we reuse this data for some other purposes apart from surveillance as quickly as possible. And also using this sequenced investment for service delivery. So we provide service delivery for our research community and also for the private sector, not only to generate the sequence, but also to do some of the basic analysis for them. Was very controversial at the beginning, but now it's working very good at Malvern. And so we're very happy about that. What to keep or what our lesson learns? Our international partnerships were key. The national database was first seated at the Wellcome Trust Sanger Institute before moving to our institute. The bioinformatics training is never enough. Now we are training our provinces, and when they go to international courses, they do have an experience by real languages. It is quite easy, for example, for people in Buenos Aires to speak English, but it's not the same in the rest of the provinces in the country. We are rethinking our IT infrastructure locally, regionally, and have to rethink this network of networks and how to link to the existing laboratory networks. So this is part of our team. This is an old picture. We are increasing 50% now of people, which for us is a lot. And here's our network. We have our first in-person meeting this year in May. So this is all the provinces plus international partners that join us. And well, now I'm very happy that our main founder is the Ministry of Health in Argentina and not the other ones. So that has been quite an improvement for now. Thanks very much.
Hi, I'm Anne-Françoise Adam from France. Thank you for your really nice presentation. So you said that the purpose of your organization is for health, etc., but also there was environment health. And I was wondering what was the exact scope of the environment part.
We do have, so under the 14 Institute, there is one institute that was created around 7 years ago, but lately repurposed to environmental surveillance. So yes, under Malvern Institute, there is a big part of environmental surveillance. All our genomic environmental projects are led by them together with us in both parts. So yes, we are seated under the Ministry of Health, but we work directly with other ministries as well. So for example, Argentina has a law on AMR, and AMR surveillance, where all the ministries are involved together. So we're quite excited about that.
In my talk earlier today, I talked about the new WHO Pandemic Accord or CA Plus agreement. And the choice there is very much a reaction to COVID, to the pandemic, and the legal scope that they're looking at is pathogens of pandemic potential. And for some, that looks like a narrow scope, but maybe that's the most important priority. And like you sort of said, going through the different, networks that exist and the different levels of development, there might be a need to sort of prioritize. But I wonder certainly for Salmonella and for E. coli, there's no pandemic potential, right? And probably not for some of the others. So what's your personal sort of experience or thinking on how important is pandemic potential, or sort of like pathogens as a class, something that we as a community need to figure out infrastructure for as a broader group? What are your personal opinions?
Well, I will move aside from the negotiations, which is completely governmental, and I'm part advisor on that. But as an Institute of Public Health, so I'm not talking about the WHO negotiation, we definitely not only focus on pandemic potential, we also do any kind of surveillance of pathogens of public health relevance, which is also include endemic diseases as well, environmental health. So for us, the scope is quite broad, and it's seated under the objective of Unleash Malbrand. So it's in the core of our mission. So not only for pandemic preparedness, which is not under the scope original from the Institute. So it's any pathogens of public health relevance.